Hello, beautiful souls. Welcome to this quiet space, a place where minds slow down and hearts rest. Before we begin this gentle, sleepy journey, just a small moment of support. If this meditation brings you calm, comfort, or clarity, please subscribe to the channel, gently tap the like button, and leave a comment to let us know you are here. Your support helps us continue creating these peaceful spaces for your mind, for your heart. Now, there is nothing else you need to do, nothing to think about. Let's begin. Close your eyes now and slowly settle into a comfortable position. Take a gentle breath in and slowly breathe out. Let your shoulders relax, let your jaw loosen, and allow your whole body to feel heavy. The human brain, which weighs approximately 3 pounds and contains roughly 86 billion neurons, operates through electrochemical signals that travel along pathways formed by repeated patterns of activation, and when psychologists speak about mental processes, they are referring to these physical events occurring within the neural tissue of the central nervous system, not to abstract phenomena existing separately from biological structure. The term mental process describes the sequence of neural firings that correspond to what we subjectively experience as thinking, remembering, perceiving, or feeling, and these processes are measurable through various neuroimaging techniques that detect changes in blood flow, electrical activity, and metabolic consumption within specific regions of brain tissue. When a thought occurs, it represents a particular configuration of neurons firing in temporal and spatial patterns, with each neuron communicating with thousands of other neurons through synapses, which are the microscopic gaps where neurotransmitters carry signals from one cell to another, and this entire system operates according to principles of biochemistry and physics rather than through any mechanism that exists outside the physical realm. Thoughts, from a neuroscientific perspective, are not experiences in the philosophical sense, but rather patterns of neural activity that can be mapped and measured with increasing precision as technology advances. And what we call a single thought is actually a distributed pattern of activation across multiple brain regions, with different areas contributing different types of processing, such as semantic content, emotional valence, sensory associations, and motor planning components. The prefrontal cortex, which occupies the front portion of the brain, behind the forehead, participates heavily in what we consider deliberate thinking, while the temporal lobes process auditory information and aspects of language comprehension, and the parietal lobes integrate sensory information from various modalities, and all of these regions communicate through white matter tracts that are bundles of axons wrapped in myelin sheaths that facilitate rapid signal transmission. The subjective experience of having a thought corresponds to a particular firing pattern that may last anywhere from milliseconds to several seconds. And this pattern involves both excitatory neurons that increase the likelihood of downstream neurons firing and inhibitory neurons that decrease such likelihood, creating a complex balance that determines which neural pathways become active at any given moment. The brain, as an organ that consumes approximately 20% of the body's total energy, despite representing only about 2% of body weight, has evolved various mechanisms to minimize metabolic expenditure while still processing the enormous quantity of information required for survival. And one of the most fundamental ways it achieves this efficiency is through the development of routines and predictable patterns that reduce the computational demands placed on neural systems. When the environment or a task is novel, the brain must allocate significant resources to processing unexpected stimuli, evaluating potential responses, and monitoring outcomes, which requires heightened activation across multiple networks, including those involved in attention, working memory, and executive control. But when the same environment or task becomes familiar through repetition, the brain can shift to more automatic processing modes that require substantially less energy and cognitive effort. This preference for routine is not a psychological quirk or a limitation of imagination, but rather an adaptive feature that allows the nervous system to reserve its limited resources for situations that genuinely require flexible, creative problem solving. Rather than expending those resources on every mundane action throughout the day, predictability in the environment reduces the need for constant vigilance and evaluation, because when patterns repeat reliably, the brain can develop internal models that anticipate what will happen next. And these predictive models allow for faster processing, with less active attention required, which is why familiar routes, familiar tasks, and familiar social interactions all feel effortless compared to novel situations that demand full conscious processing. The basal ganglia, a group of structures located deep within the brain, play a particularly important role in habit formation and the execution of learned motor sequences. And as behaviors are repeated, the neural circuits representing those behaviors become increasingly refined and automatic, requiring progressively less involvement from the prefrontal cortex and other areas associated with deliberate control. This shift from controlled to automatic processing can be observed in studies measuring brain activity during skill acquisition, where early stages of learning show widespread activation across many brain regions, but after extensive practice, activity becomes concentrated in more specific circuits that can execute the learned behavior with minimal metabolic cost and minimal conscious oversight. Repetition serves a calming function within neural systems because it reduces uncertainty. And uncertainty is metabolically expensive for the brain to process, since it requires maintaining multiple possible interpretations or response options simultaneously, until additional information resolves which interpretation or response is correct. Whereas repeated exposure to the same stimulus or situation allows the brain to settle into a single, well-established pattern of response that has been validated through prior experience. The amygdala, which is often discussed in relation to fear and emotional processing, shows decreased activation when stimuli are familiar compared to when they are novel. And this reduction in amygdala activity corresponds to a decrease in the physiological arousal that accompanies uncertainty and potential threat. When an environment contains predictable elements that repeat in regular patterns, the brain's threat detection systems can essentially stand down from their monitoring functions, allocating fewer resources to scanning for danger and more resources to other processes, or to rest and recovery. And this reallocation is experienced subjectively as a sense of ease or comfort that people often describe when they are in familiar surroundings, engaging in familiar activities. The reduction of cognitive effort that accompanies familiarity is not simply a matter of conscious ease, but involves measurable changes in neural efficiency with familiar stimuli processed through more streamlined pathways that require fewer synaptic connections to achieve recognition and appropriate response. And this streamlining occurs through a process called synaptic pruning, where connections that are rarely used gradually weaken and eventually disappear, while connections that are frequently activated become stronger through mechanisms involving protein synthesis and structural changes at the synaptic level. The principle of neural efficiency suggests that the brain continuously optimizes its circuitry to minimize the total energy required to perform necessary functions. And this optimization naturally favors repetition and routine because novel processing inherently involves more widespread activation, more trial and error testing of potential responses, and more metabolic expenditure per unit of behavior or cognition produced. Breathing, which is controlled by respiratory centers in the brainstem and normally proceeds without conscious attention, can be voluntarily modified. And when breathing is slowed deliberately, this alteration affects the autonomic nervous system through several interconnected mechanisms involving both neural and chemical signaling pathways. The autonomic nervous system consists of two main branches: the sympathetic nervous system, which generally increases arousal and prepares the body for activity, and the parasympathetic nervous system, which generally decreases arousal and supports rest, digestion, and recovery. And these two branches work in opposition to maintain what is called autonomic balance. Slow breathing, particularly when the exhalation phase is prolonged relative to the inhalation phase, stimulates the vagus nerve, which is the 10th cranial nerve and the primary nerve of the parasympathetic nervous system, carrying signals from the brainstem to the heart, lungs, and digestive organs. And increased vagal activity leads to a reduction in heart rate, a decrease in blood pressure, and a shift in overall autonomic tone toward parasympathetic dominance. The relationship between breathing rate and autonomic tone operates through multiple pathways, including mechanical stretch receptors in the lungs that send signals to the brainstem, chemical sensors that detect changes in blood oxygen and carbon dioxide levels, and neural circuits that link respiratory rhythm with cardiovascular regulation. And when breathing slows, these various pathways all contribute to a cascade of physiological changes that collectively reduce arousal and promote a state of decreased activation across multiple body systems. The heart rate typically shows a phenomenon called respiratory sinus arrhythmia, where the heart beats slightly faster during inhalation and slightly slower during exhalation. And this variation reflects the direct influence of breathing on cardiac regulation through vagal pathways. And when breathing is slow and regular, this rhythmic variation in heart rate becomes more pronounced and more regular, which is associated with improved autonomic function and decreased stress-related activation. The chemical effects of slow breathing include mild increases in carbon dioxide levels in the blood, which may seem counterintuitive since carbon dioxide is often thought of as a waste product. But modest elevations in carbon dioxide have a relaxing effect on smooth muscle tissue in blood vessels, leading to vasodilation and improved blood flow. And carbon dioxide also affects the pH of blood and cerebrospinal fluid, which influences neural excitability through effects on ion channels and neurotransmitter function. Attention, which neuroscientists define as the selective allocation of cognitive resources to particular stimuli or tasks, while filtering out irrelevant information, is not a stable state that can be maintained indefinitely, but rather a dynamic process that fluctuates over time. According to the availability of metabolic resources and the intensity of competing demands on neural systems, the networks that support sustained attention include regions of the prefrontal cortex, the parietal cortex, and subcortical structures such as the thalamus, which acts as a relay station for sensory information traveling to the cortex. And these networks must be actively maintained through ongoing neural firing that consumes glucose and oxygen delivered through cerebral blood flow. When there are no external stimuli demanding attention, or no internal goals requiring focused processing, these attention networks naturally reduce their activity level, and attention begins to drift, in a process that is not a failure of mental discipline, but rather a default mode of operation that allows the brain to conserve energy when sustained focus is not required by circumstances. The default mode network, which is a set of brain regions that shows increased activity during rest and decreased activity during externally focused tasks, becomes dominant when attention is not directed toward specific external stimuli or deliberate cognitive tasks. And this network includes the medial prefrontal cortex, the posterior cingulate cortex, the precuneus, and portions of the temporal lobes. And activity in this network is associated with spontaneous thought, autobiographical memory retrieval, and what researchers sometimes call stimulus-independent thought. When the mind wanders, activity shifts from networks supporting external attention to this default mode network. And the content of mind wandering typically includes memories, future planning, social scenarios, and self-referential processing. Though much of this activity occurs below the threshold of full conscious awareness, and may be experienced simply as a vague sense of mental drift, rather than as discrete thoughts with clear content. The wandering of the mind in the absence of stimulation is an energy-conserving state, because it involves lower levels of metabolic activity compared to focused attention on complex tasks. And this wandering occurs more readily when the environment provides minimal sensory input. or when sensory input is highly repetitive and predictable, requiring little processing to interpret or respond appropriately. The brain naturally seeks equilibrium between different modes of operation, alternating between periods of focused external attention and periods of internally directed processing or diffuse awareness. And this alternation follows patterns that are partly governed by circadian rhythms, partly influenced by homeostatic sleep pressure that builds during waking and dissipates during sleep, and partly responsive to the specific demands and opportunities present in the current environment. Monotonous environments with low variability in sensory input promote mind wandering, because there is insufficient novelty or complexity to sustain engagement of the attention networks, and the brain defaults to its lower energy resting state. When external circumstances do not require otherwise, the tendency of the brain to simplify information is evident in numerous perceptual and cognitive processes. From the way visual stimuli are processed through increasingly abstract representations, as signals move from the retina through the lateral geniculate nucleus of the thalamus to the primary visual cortex, and then to higher-order visual areas, to the way language comprehension extracts meaning from complex grammatical structures, without retaining the exact wording of each sentence. Simplification reduces the amount of information that must be actively maintained in working memory, and reduces the number of distinctions that must be tracked when making decisions or forming judgments. And this reduction in complexity serves the fundamental goal of metabolic efficiency. By limiting the scope of neural activation required to achieve adequate functioning in most situations, the brain uses various heuristics and categorical representations to compress detailed sensory input into manageable summaries. And while these simplifications sometimes lead to errors or biases, they generally provide sufficient accuracy for practical purposes, while requiring far less processing than would be needed to maintain complete fidelity to all available information. Categorical perception is one example of this simplification process, where continuous variation in sensory stimuli is perceived as belonging to discrete categories, with sharp boundaries between them, such as the way colors are perceived as distinct hues, even though the physical spectrum of light wavelengths varies continuously, or the way phonemes in speech are heard as distinct sounds, even though the acoustic properties of speech vary smoothly over time. This categorical organization allows the brain to apply learned associations and behavioral responses at the category level, rather than having to evaluate each individual instance as a unique case, which would be computationally prohibitive given the enormous number of distinct stimuli encountered throughout a day. The process of chunking in memory is another manifestation of this simplification tendency, where individual pieces of information are grouped together into larger units that can be treated as single items for the purposes of storage and retrieval, such as remembering a phone number as several multi-digit chunks, rather than as a sequence of individual digits. And this chunking dramatically increases the effective capacity of working memory by reducing the number of discrete elements that must be maintained. The nervous system regulates arousal levels through a complex interplay of neurotransmitter systems, hormonal signals, and distributed neural networks that collectively determine the overall level of alertness, responsiveness, and physiological activation at any given time. And this regulation operates automatically without requiring conscious intervention or decision-making. The ascending reticular activating system, which originates in the brainstem and projects widely throughout the cortex, plays a central role in maintaining wakefulness and modulating arousal, releasing neurotransmitters such as norepinephrine, dopamine, serotonin, and acetylcholine that influence the excitability of neurons across the entire brain. The locus coeruleus, a small nucleus in the brainstem containing norepinephrine-producing neurons, is particularly important for regulating arousal in response to salient stimuli. And activation of the locus coeruleus increases vigilance and responsiveness, while decreased activity in this structure is associated with reduced arousal and the transition towards sleep. The regulation of arousal follows principles of homeostasis, where deviations from optimal levels trigger compensatory responses that restore balance. And this homeostatic regulation ensures that arousal levels remain within functional ranges, rather than escalating to extremes or dropping to levels incompatible with necessary activities. When arousal has been elevated for extended periods due to sustained demands or stressors, homeostatic mechanisms promote a subsequent period of reduced arousal to allow recovery. And this compensatory reduction is experienced as fatigue, a desire to rest, or a difficulty maintaining effort on demanding tasks. The hypothalamus, which regulates numerous homeostatic processes, including temperature, hunger, thirst, and sleep-wake cycles, integrates information about current arousal levels with information about circadian timing and metabolic state to coordinate appropriate adjustments in arousal through both neural and hormonal pathways. Silence and low levels of environmental stimulation reduce cognitive load because they minimize the amount of sensory information that must be processed and filtered by attentional systems. And this reduction in sensory input allows more of the brain's limited processing capacity to be allocated to whatever task is being performed, or if no task is being performed, allows neural systems to enter lower activity states that support recovery and metabolic conservation. Cognitive load, refers to the total amount of mental effort being used in working memory at a given time. And this load increases with the complexity of information being processed. The number of distinct elements that must be tracked simultaneously, and the amount of interference from irrelevant stimuli that must be actively suppressed or ignored. In environments with high levels of sensory stimulation, particularly stimulation that is unpredictable, or that includes elements resembling potentially important signals such as speech or sudden noises, the brain must allocate resources to monitoring and filtering this input. Even when the person is attempting to focus on an unrelated task. And this monitoring consumes resources that would otherwise be available for the focal task or for rest. The prefrontal cortex, which supports working memory and executive functions such as planning and cognitive control, shows reduced activity in conditions of low stimulation. And this reduction is associated with decreased subjective effort and decreased metabolic demands on neural tissue in these regions. Functional neuroimaging studies comparing brain activity during rest in quiet environments versus rest in environments with auditory or visual stimulation show that stimulus-rich environments maintain higher baseline levels of activation across sensory cortices and attention networks, even when no specific task is being performed, indicating that the mere presence of stimulation imposes a processing burden, regardless of whether that stimulation is actively attended to or consciously noticed. The benefits of silence and low stimulation are particularly evident when measuring recovery from mental fatigue, where individuals who rest in quiet, low-stimulation environments show faster restoration of performance on attention-demanding tasks, compared to individuals who rest in environments with television, music, or conversation present as background stimulation. Memory contributes to emotional neutrality through a process called emotional habituation, where repeated exposure to a stimulus leads to progressively weaker emotional responses, even though the stimulus itself remains unchanged. And this habituation represents a form of learning, where the brain updates its predictions about the significance and consequences of stimuli based on accumulated experience. When a stimulus is first encountered, particularly if it is intense or unexpected, it activates emotional processing systems, including the amygdala and related structures. And these systems trigger physiological arousal and behavioral responses appropriate to potential threats or opportunities. However, if this stimulus is encountered repeatedly, without significant positive or negative consequences, the emotional response gradually diminishes through a combination of mechanisms, including decreased amygdala responsiveness, increased inhibitory control from prefrontal regions and altered neurotransmitter signaling that reduces the gain of emotional processing systems. The hippocampus, which is essential for forming new episodic memories, works in conjunction with the amygdala to associate emotional significance with specific events and contexts. And the strength of emotional memories depends on the level of arousal present during encoding, with high arousal events producing stronger and more persistent memories than low arousal events. Over time, as memories are repeatedly retrieved and reconsolidated, their emotional components tend to fade more rapidly than their factual content, a process that allows people to remember what happened without re-experiencing the full intensity of the original emotional response. And this separation of emotional and factual components of memory is thought to involve changes in connectivity between the hippocampus, the amygdala, and cortical regions where long-term memories are ultimately stored. The emotional neutrality of well-established memories means that familiar stimuli and situations, even if they were initially arousing or significant, become less capable of triggering strong emotional responses. And this contributes to the calming effect of routine and familiarity. By reducing the frequency and intensity of emotional activation throughout daily life, the brain filters the vast majority of sensory information arriving at receptor organs, allowing only a small fraction to reach conscious awareness. And this filtering is essential because the total amount of sensory data arriving through visual, auditory, tactile, olfactory, and gustatory channels far exceeds the processing capacity of higher-level cognitive systems. The retina alone contains approximately 130 million photoreceptors that transduce light into neural signals. But the optic nerve contains only about 1 million axons, carrying information from the retina to the brain, indicating that substantial compression and filtering must occur even in the earliest stages of visual processing, before information reaches cortical areas. This compression continues at each successive stage of processing, with lateral inhibition enhancing contrast and reducing redundancy. Receptive fields extracting increasingly abstract features, such as edges and orientations, and attention mechanisms selectively amplifying signals from relevant locations or features, while suppressing signals from irrelevant sources. The filtering of sensory information operates largely automatically through circuits that have been shaped by evolution to prioritize stimuli with survival relevance, such as sudden movements, high contrast boundaries, and sounds with characteristics resembling vocalizations or alarm calls. And through learning mechanisms that adjust filtering based on individual experience and current goals. The thalamus acts as a critical filtering station, where ascending sensory signals are modulated by descending projections from the cortex. And this bidirectional interaction allows top-down expectations and priorities to influence which sensory information passes through to cortical processing, and which information is attenuated or blocked at the thalamic level. The extensive filtering of sensory input means that conscious experience represents a highly selective sample of available information, rather than a complete record. And this selectivity is necessary to prevent cognitive systems from being overwhelmed by the continuous flood of sensory data generated by receptor organs. Habits reduce decision-making effort by establishing automatic links between specific contexts and specific behavioral responses, such that when the context is encountered, the corresponding behavior is initiated without requiring deliberate evaluation of options or conscious choice among alternatives. The formation of habits involves gradual strengthening of associations in the basal ganglia, particularly in the dorsolateral striatum, which receives inputs from sensory and motor cortex, and sends outputs through the globus pallidus and thalamus back to motor areas, creating loops that can execute learned sequences of actions with minimal involvement of prefrontal decision-making systems. Initially, when a behavior is new, its execution requires substantial prefrontal activity to maintain goals in working memory, monitor performance, and adjust actions based on feedback. But as the behavior is repeated in consistent contexts, control gradually transfers from prefrontal cortex to striatal circuits that can operate more efficiently and automatically. The automaticity of habits means that they can be executed while conscious attention is directed elsewhere, which is why people can engage in complex habitual behaviors, such as driving familiar routes, while simultaneously holding conversations or thinking about unrelated topics. And this parallel processing is possible because habitual behaviors rely on different neural circuits than those required for deliberate, novel problem solving. The reduction in decision-making effort provided by habits has significant implications for mental energy expenditure throughout the day, because each decision, even a simple one, requires activation of prefrontal networks and consumes metabolic resources, and the cumulative effect of hundreds of small decisions can lead to a state called decision fatigue, where the quality of decisions deteriorates and the subjective effort required for deciding increases. By converting frequently performed behaviors into automatic habits, the brain reduces the total number of decisions required, preserving cognitive resources for situations where flexible decision-making is genuinely necessary. Predictable environments feel mentally quiet because they minimize the need for active monitoring, evaluation, and response preparation, allowing neural systems to settle into stable patterns of low-level activity, rather than maintaining the heightened activation required to deal with uncertainty and change. Environmental predictability can come from physical regularities, such as consistent spatial layouts, stable lighting, and temperature, an absence of sudden changes in sensory input, or from temporal regularities, such as consistent daily schedules and routines that allow accurate anticipation of what will happen and when. And both forms of predictability contribute to reduced cognitive demands and reduced physiological arousal. The subjective experience of mental quietness in predictable environments corresponds to measurable decreases in activation across attention networks, executive control networks, and emotional processing systems. And this decreased activation is associated with lower heart rate, lower blood pressure, reduced muscle tension, and decreased secretion of stress hormones such as cortisol. The relationship between environmental predictability and mental quietness is evident in studies examining physiological stress responses, where individuals in highly variable, unpredictable environments show chronically elevated cortisol levels and higher baseline activation of the sympathetic nervous system compared to individuals in stable, predictable environments, even when the actual demands and challenges are similar between the two groups. The difference lies not in the objective difficulty of tasks or situations, but in the predictability of when demands will occur and what form they will take. Because predictability allows the brain to prepare appropriate responses in advance and to allocate resources efficiently, while unpredictability requires maintaining higher baseline levels of readiness and vigilance to deal with whatever might occur. The preference for predictable environments is not culturally learned, but reflects fundamental properties of neural information processing, where prediction errors, which occur when actual input differs from expected input, drive learning and adaptation, but also require additional processing and metabolic expenditure to resolve. Perception and energy efficiency are linked through the principle that the brain continuously attempts to minimize the total prediction error across all sensory modalities, while using the minimum amount of neural activity necessary to achieve this minimization. And this principle underlies theories of perception that view the brain as a prediction machine that generates internal models of the world and continuously updates these models based on incoming sensory evidence. When sensory input matches predictions generated by internal models, very little neural activity is required because the prediction error is zero or near zero, and neural circuits can remain in low activity states that conserve energy. But when sensory input deviates from predictions, prediction error signals are generated and propagated through cortical hierarchies, triggering increased neural activity to update models and reduce future prediction errors. This framework explains why familiar, expected stimuli are processed with less neural activity than novel, unexpected stimuli. Why attention tends to be automatically captured by deviations from established patterns, and why predictable, repetitive environments are experienced as less demanding than variable, surprising environments. The visual system provides clear examples of energy-efficient perceptual processing, where mechanisms such as center surround receptive fields implement efficient coding strategies that transmit information about spatial changes, while filtering out uniform or slowly varying regions that can be accurately predicted from surrounding context, and where higher-level areas extract statistical regularities such as common shapes, textures, and patterns that allow compression of visual information into compact representations requiring fewer neurons to maintain. The auditory system similarly uses predictive processing to anticipate upcoming sounds based on temporal patterns in the recent past. And when sounds conform to these predictions, they are processed efficiently with minimal surprise signals, while sounds that violate predictions trigger enhanced processing to determine the source and significance of the unexpected input. Across all sensory modalities, the nervous system appears to be organized to achieve perceptual accuracy with minimal metabolic cost. And this organization naturally leads to preferences for regularity, predictability, and familiarity that reduce the total amount of prediction error that must be processed. Rest is a biological process involving specific changes in neural activity, neurotransmitter systems, and cellular metabolism, rather than simply the absence of activity or a reward earned through effort. And the need for rest accumulates according to homeostatic mechanisms that track the duration and intensity of prior waking activity and generate increasing pressure to enter restorative states as this debt accumulates. Sleep, which is the most complete form of rest, involves a shift from wake-promoting neurotransmitter systems, including norepinephrine, serotonin, dopamine, and histamine, to sleep-promoting systems involving gamma-aminobutyric acid, or GABA, and adenosine. And this shift is coordinated by circuits in the hypothalamus and brainstem that function as a flip-flop switch, maintaining either stable wakefulness or stable sleep, and resisting intermediate states. The accumulation of adenosine in the brain during waking is one key signal promoting sleep, as adenosine is a byproduct of ADP breakdown, which is the primary energy currency of cells. And elevated adenosine levels inhibit wake-promoting neurons, while exciting sleep-promoting neurons, creating increasing homeostatic pressure for sleep that can only be relieved by actually sleeping. During sleep, particularly during slow-wave sleep, which is characterized by large amplitude, low-frequency oscillations in cortical electrical activity, the brain undergoes processes that cannot occur during waking, including clearance of metabolic waste products through the glymphatic system, consolidation of memories through selective strengthening and weakening of synaptic connections, and restoration of neurotransmitter stores that were depleted during waking. The glymphatic system, which was only discovered in recent years, involves flow of cerebrospinal fluid through spaces between brain cells, carrying away proteins and metabolites that accumulate during neural activity. And this flow increases dramatically during sleep compared to waking, suggesting that one essential function of sleep is to allow this cleaning process to operate efficiently. The energy demands of the brain decrease during sleep compared to waking, but the brain remains quite active during sleep, consuming only slightly less glucose and oxygen than during quiet wakefulness, which indicates that sleep is not merely a shutdown of neural activity, but rather a shift to different patterns of activity that support different functions. Mental fatigue, which develops during sustained cognitive effort, differs from physical fatigue in that it primarily reflects depletion of metabolic resources in specific neural circuits and accumulation of byproducts that interfere with efficient neural signaling, rather than exhaustion of muscle glycogen stores or accumulation of lactate in muscle tissue. The prefrontal cortex appears particularly vulnerable to mental fatigue, showing decreased activation and decreased functional connectivity with other brain regions as sustained cognitive effort continues. And this decreased function manifests behaviorally as reduced ability to sustain attention, increased distractibility, decreased working memory capacity, and impaired decision-making and self-control. The mechanisms underlying mental fatigue likely include depletion of glucose and oxygen locally in active neural tissue. Faster than blood flow can replenish these resources. Accumulation of adenosine and other metabolic byproducts that modulate neural excitability, and alterations in neurotransmitter availability, particularly in dopamine and norepinephrine systems, that support motivation and sustained effort. Mental fatigue is not simply a subjective feeling, but can be objectively measured through changes in task performance, changes in brain activity patterns, and changes in physiological markers, such as heart rate variability and cortisol levels. And these objective measures show that mental fatigue develops progressively during sustained cognitive demands. Even when people report feeling capable of continuing, suggesting that some aspects of fatigue operate below conscious awareness. To protect neural systems from damage that could result from excessive depletion of energy stores or excessive accumulation of metabolic waste. Recovery from mental fatigue requires a period of reduced cognitive demand to allow restoration of depleted resources and clearance of accumulated byproducts. And this recovery occurs more rapidly in conditions of low environmental stimulation, where the brain can enter low-activity resting states, rather than having to continue processing sensory input and maintaining vigilance, even while attempting to rest. The brain naturally seeks balance rather than intensity, because extreme activation in either direction, whether high arousal or deep suppression, is metabolically expensive to maintain and potentially damaging to neural circuits if sustained for prolonged periods. And the regulatory systems governing arousal, emotion, and cognitive effort all include negative feedback mechanisms that resist movement away from moderate baseline levels and promote return to those baselines after perturbations. This tendency toward balance can be seen in the way emotional responses naturally decay over time, even when the triggering situation persists, in the way arousal levels fluctuate within a bounded range, rather than escalating without limit, and in the way cognitive performance shows optimal levels of task difficulty that are neither too easy nor too difficult, with performance degrading when demands fall outside this optimal zone. The concept of homeostasis applies not only to physiological variables such as body temperature and blood glucose, but also to psychological and neural states, where set points exist for optimal levels of stimulation, challenge, and arousal. And deviations from these set points trigger compensatory responses that restore balance. The seeking of balance rather than intensity is evident in the way people spontaneously alternate between different activities and mental states throughout the day, shifting from focused work to brief periods of rest or mind wandering, from social interaction to solitude, from physical activity to sedentary rest. And these spontaneous shifts reflect homeostatic regulation that prevents any single state from being maintained so long that resources become depleted or systems become dysregulated. The preference for moderate levels of stimulation and arousal is also seen in aesthetic preferences, where people generally prefer intermediate levels of complexity, novelty, and intensity in music, visual art, and other forms of sensory stimulation, finding very simple, predictable patterns boring, but finding very complex, unpredictable patterns unpleasant or overwhelming. And this preference for intermediate levels optimizes the balance between engagement, which requires some degree of unpredictability to sustain attention, and processing efficiency, which benefits from predictability and familiarity. Psychological adaptation occurs gradually through incremental changes in neural circuitry, neurotransmitter sensitivity, and hormonal regulation, rather than through sudden transformations. And this gradual nature of adaptation reflects the fact that neuroplasticity, which is the capacity for neural circuits to modify their structure and function in response to experience, operates through molecular and cellular processes that require time to unfold. Synaptic plasticity, which is the strengthening or weakening of connections between neurons, based on patterns of correlated activity, follows rules such as long-term potentiation and long-term depression that require repeated activation over minutes to hours to produce lasting changes. And these changes then stabilize over days to weeks through processes involving protein synthesis, structural modifications to synaptic connections, and alterations in gene expression. The consolidation of new learning into stable long-term memory follows a similar time course, with initial learning producing fragile memory traces that are susceptible to interference or forgetting. But repeated practice and time allow these traces to become progressively more stable and resistant to disruption. Adaptation to changed environments or circumstances proceeds through similar, gradual processes, where initial responses to change involve activation of stress systems and deployment of cognitive resources to develop new behavioral strategies. And these responses gradually normalize as the new situation becomes familiar and routines develop that reduce the cognitive demands of navigating the changed context. Studies of adaptation to major life changes, such as relocation, job changes, or relationship transitions, show that emotional responses and stress markers typically peak during the first weeks or months after the change, and then gradually decline toward baseline levels over subsequent months, even when objective circumstances remain different from what they were before. And this decline reflects genuine neural and physiological adaptation, rather than mere suppression or denial of reactions. The time course of adaptation varies depending on the magnitude of change and individual differences in plasticity and coping resources. But the general pattern of gradual adjustment is consistent across different types of challenges and different populations. Calm states, from a neuroscientific perspective, are not special states requiring unusual conditions or techniques to achieve, but simply reflect low levels of activity in systems related to arousal, threat detection, and cognitive effort, combined with relative dominance of parasympathetic nervous system activity over sympathetic activity. And these states occur naturally when environmental demands are low, when there are no threats or stressors requiring attention, and when metabolic resources are adequate to meet current needs without strain. The default mode network activity that characterizes rest and relaxed wakefulness shows a pattern of slow, synchronized oscillations across distributed cortical regions. And this synchronized activity is thought to support maintenance and updating of internal models of self and world, while consuming less energy than the more rapid, desynchronized activity patterns associated with active task performance. The neurotransmitter GABA, which is the primary inhibitory neurotransmitter in the brain, plays a crucial role in generating calm states. By reducing the excitability of neural circuits and preventing runaway, positive feedback that could lead to excessive activation, and drugs that enhance GABA signaling, such as benzodiazepines and certain anesthetics, produce sedation and anxiolysis by amplifying these natural inhibitory mechanisms. Calm states are associated with increased heart rate variability, which is the beat-to-beat variation in heart rate that reflects parasympathetic influence on cardiac pacing. And higher heart rate variability is generally considered a marker of better autonomic function and greater physiological resilience, because it indicates that the cardiovascular system can flexibly adjust to changing demands, rather than being locked into rigid patterns. The breathing pattern during calm states typically becomes slower and more regular, with prolonged exhalations that stimulate vagal activity and reinforce parasympathetic dominance, creating a positive feedback loop where slow breathing promotes calm and calm promotes slow breathing. Muscle tension decreases during calm states, as motor neurons receive less excitatory input from upper motor centers. And this decreased tension is particularly evident in muscles of the face, neck, and shoulders that are sensitive to stress and arousal. And the reduction in muscle tension both reflects and contributes to the overall decrease in physiological activation that characterizes calm states. The ordinariness of calm states, their status as default modes of neural operation when specific demands are absent, means that cultivating calm does not require learning exotic practices or achieving rare psychological conditions, but rather involves removing or reducing factors that prevent the natural settling of neural systems into low-activity configurations. Factors such as excessive environmental stimulation, chronic stressors that maintain activation of threat detection systems, sleep deprivation that interferes with normal homeostatic regulation, or consumption of substances such as caffeine that pharmacologically promote arousal by blocking adenosine receptors and stimulating release of excitatory neurotransmitters. When these interfering factors are minimized, the brain's intrinsic regulatory mechanisms naturally guide the system toward balanced states, characterized by moderate arousal, efficient processing, and absence of unnecessary activation. And these states feel subjectively calm, not because of any special quality they possess, but simply because they lack the tension, urgency, and effortfulness that characterize states of elevated arousal or sustained cognitive demand. The neural correlates of subjective calmness include reduced activity in the anterior cingulate cortex, which is involved in conflict monitoring and error detection, reduced activity in the insula, which processes interoceptive signals related to bodily states and emotional feelings, and reduced activity in the amygdala and related limbic structures involved in emotional reactivity and threat assessment, while activity in the prefrontal cortex may remain moderate, but shifts toward more medial regions associated with self-referential processing, and away from lateral regions associated with executive control and working memory. These patterns of reduced activation are not states of neural shutdown, but rather reflect a different configuration of ongoing activity, where processing is biased toward internally generated patterns rather than externally driven responses, where predictive models dominate over correction of prediction errors, and where tonic, steady-state activity replaces the rapid fluctuations associated with active task engagement. The physiological state associated with subjective calmness includes not only decreased sympathetic and increased parasympathetic tone, but also changes in the hypothalamic pituitary adrenal axis that regulates stress hormone secretion, with calm states characterized by lower circulating cortisol levels and flatter diurnal cortisol rhythms that follow the natural circadian pattern of high morning levels that decline through the day, rather than showing the elevated and irregular patterns associated with chronic stress. Cortisol has complex effects on brain function, with moderate levels supporting memory formation and cognitive performance, but elevated levels impairing prefrontal function and hippocampal plasticity. And chronically elevated cortisol is associated with structural changes in the brain, including reduced volume of the hippocampus and prefrontal cortex, and increased volume of the amygdala, changes that have functional consequences for emotional regulation and stress reactivity. The reduction in cortisol levels during calm states both reflects and supports the decreased activation of threat-related neural systems, creating conditions favorable for recovery and restoration. Perception of time is altered in calm states, compared to states of high arousal or intense cognitive engagement, with time typically seeming to pass more quickly during calm than during stress or boredom. And this difference in subjective time perception likely reflects differences in the density of attention-demanding events that are processed and encoded into memory. Because the subjective duration of a period is partly determined by how much mental activity occurred during that period, with event-rich periods seeming longer in retrospect than event-poor periods, even though the actual duration was the same. During calm states, with low external stimulation and minimal cognitive demands, fewer discrete events are encoded, resulting in sparse memory records and compressed subjective duration. While during aroused states or novel situations, heightened attention leads to encoding of more details and more discrete events, creating dense memory records that make the period seem longer when recalled. This relationship between arousal, attention, memory encoding, and time perception is part of why familiar, routine activities seem to pass quickly, while novel experiences or stressful situations seem to stretch time, and why periods of rest or relaxation may seem to end sooner than expected, even though they may provide more recovery than longer periods of low-quality rest interrupted by demands or distractions. The default mode network activity that characterizes restful states shows characteristic patterns of connectivity between regions, including the medial prefrontal cortex, which is involved in self-related processing and social cognition, the posterior cingulate cortex and precuneus, which are involved in autobiographical memory and spatial processing, and the medial temporal lobes, including the hippocampus, which support episodic memory. And these regions show correlated activity during rest that is thought to support maintenance and consolidation of memories, and integration of new experiences with existing knowledge structures. The anti-correlation between default mode network activity and activity in networks supporting external attention and executive control means that as one system becomes more active, the other becomes less active. And this reciprocal relationship may reflect a fundamental trade-off in how the brain allocates its limited processing resources between internal mentation and external engagement. During calm, restful states, the balance shifts toward default mode activity and away from externally focused networks, allowing spontaneous thought processes to unfold without interruption from environmental demands or deliberate cognitive tasks. Sleep architecture, which refers to the cyclical progression through different sleep stages throughout the night, shows characteristic patterns of NREM or non-rapid eye movement sleep alternating with REM or rapid eye movement sleep in cycles lasting approximately 90 minutes, with NREM sleep dominating the early part of the night and REM sleep becoming more prominent in later cycles. NREM sleep is further divided into lighter stages N1 and N2, and deeper stage N3, also called slow-wave sleep. And these stages are defined by characteristic patterns of electrical activity recorded from the scalp using electroencephalography, with slow-wave sleep showing the largest amplitude, lowest frequency oscillations, and representing the deepest and most restorative stage of sleep. The functions of different sleep stages appear to be partially distinct, with slow-wave sleep particularly important for metabolic restoration and memory consolidation of declarative information, while REM sleep is associated with emotional processing and consolidation of procedural memories, and disruption of specific sleep stages produces selective deficits in the functions those stages support. The transition from wakefulness to sleep proceeds through gradual changes in neural activity rather than abrupt switches, with the first stage N1 representing a transitional state, where wakefulness and sleep alternate in brief epochs, and awareness of the environment becomes intermittent and unreliable, then progressing to stage N2, where sleep becomes more consolidated, though still relatively light and easily interrupted by noise or other disturbances. And finally, to slow-wave sleep, where arousal threshold is highest and sensory processing is most suppressed. The neural mechanisms that generate slow waves during deep sleep involve synchronized alternation of cortical neurons between depolarized up states, where neurons are firing, and hyperpolarized down states, where neurons are silent. And this alternation occurs simultaneously across large populations of neurons, creating the large amplitude oscillations visible in EEG recordings. And these oscillations are thought to support synaptic homeostasis by allowing widespread synaptic depression that counteracts the net synaptic strengthening that occurs during waking learning and experience. The circadian clock, which is located in the suprachiasmatic nucleus of the hypothalamus and consists of approximately 20,000 neurons, with intrinsic rhythmicity based on interlocking transcriptional translational feedback loops, coordinates daily rhythms in sleep-wake behavior, core body temperature, hormone secretion, and numerous other physiological and behavioral variables. And this clock is synchronized to the external light-dark cycle, primarily through direct input from specialized photoreceptive ganglion cells in the retina that detect ambient light levels and convey this information to the suprachiasmatic nucleus. The circadian promotion of sleep follows a rhythm approximately opposite to the core body temperature rhythm, with sleep propensity highest when core temperature is declining, or at its minimum during biological night, and lowest when temperature is rising, or at its maximum during biological day. And this circadian signal combines with homeostatic sleep pressure to determine the timing and quality of sleep across the 24-hour day. Disruption of circadian rhythms through shift work, jet lag, or irregular sleep schedules interferes with the normal coordination between circadian and homeostatic sleep regulation, and can lead to chronic sleep difficulties, excessive daytime sleepiness, and increased risk for various health problems, including metabolic disorders and mood disturbances. Neurotransmitter systems show characteristic circadian and sleep-wake dependent variations, with wake-promoting systems including norepinephrine from the locus coeruleus, serotonin from the raphe nuclei, histamine from the tuberomammillary nucleus, and orexin from the lateral hypothalamus, all showing high activity during waking and low activity during sleep, while sleep-promoting systems involving GABA and galanine show the opposite pattern. And these systems mutually inhibit each other, creating the flip-flop dynamics that produce stable waking and stable sleep states, with rapid transitions between them, rather than prolonged intermediate states. Adenosine, which accumulates during waking and promotes sleep, acts partly by inhibiting wake-promoting neurons, and partly by exciting sleep-promoting neurons. And caffeine produces its alerting effects by blocking adenosine receptors, and thereby removing adenosine's inhibitory influence on wake-promoting systems. Though this blockade does not actually reduce the underlying homeostatic sleep debt. And when caffeine is metabolized or wears off, the accumulated sleep pressure reasserts itself, often producing a rebound of sleepiness. The cognitive functions impaired by sleep deprivation include sustained attention, working memory, decision-making, impulse control, and emotional regulation. And these impairments reflect dysfunction, particularly of prefrontal cortex, which is especially vulnerable to sleep loss, showing decreased metabolic activity and altered functional connectivity after even moderate sleep restriction. The subjective sleepiness experienced during sleep deprivation corresponds imperfectly to the degree of objective cognitive impairment, with people often underestimating their level of impairment, particularly after chronic partial sleep restriction, where homeostatic sleep debt accumulates. But individuals adapt to their decreased functioning and may not recognize how impaired they have become compared to their fully rested baseline. Sleep deprivation also affects emotional reactivity, with sleep-deprived individuals showing increased amygdala responses to negative stimuli, and decreased functional connectivity between amygdala and prefrontal regions that normally regulate emotional responses, resulting in heightened negative emotionality and decreased ability to modulate emotional reactions through cognitive control. The restoration that occurs during sleep cannot be fully replaced by quiet rest while awake, indicating that sleep involves active processes beyond simple disengagement from environmental demands. And these active processes include the previously mentioned glymphatic clearance of metabolic waste, the synaptic homeostasis that counteracts runaway synaptic strengthening, the memory consolidation that selectively strengthens task-relevant synapses while pruning less important connections, and the hormonal regulations, including growth hormone secretion, which peaks during slow-wave sleep and supports protein synthesis and tissue repair. The specific molecular and cellular events that constitute sleep's restorative functions are still being elucidated, but appear to include upregulation of genes involved in protein synthesis and membrane trafficking, increased autophagy and cellular repair mechanisms, restoration of glycogen stores that were depleted during waking, and rebalancing of neurotransmitter systems that became dysregulated during extended waking. Individual differences in sleep need, which can range from less than 6 hours to more than 9 hours per night, For healthy adults to feel fully rested and function optimally. Reflect genetic variations in sleep regulation, including polymorphisms in genes involved in circadian rhythms, adenosine signaling, and sleep stage generation. And these differences are stable traits rather than preferences or habits that can be substantially modified through behavioral interventions. Though optimal sleep duration can be influenced by environmental demands, stress levels, and health status, the consequences of chronic insufficient sleep relative to individual need include not only the acute impairments in attention and cognition, but also longer-term health risks, including increased incidence of cardiovascular disease, metabolic syndrome and diabetes, obesity, weakened immune function, and mood disorders. And these health effects likely reflect the cumulative impact of inadequate time for the restorative processes that occur during sleep to fully complete their functions. The relationship between sleep and learning is bidirectional, with learning experiences during waking influencing subsequent sleep architecture, including increased slow-wave activity and sleep spindle density in regions that were active during learning, and sleep in turn supporting consolidation of recently learned information through reactivation of neural patterns that were active during initial encoding. Sleep-dependent memory consolidation is not simply passive protection of fragile memory traces from interference, but involves active reorganization and integration of new memories with existing knowledge structures, extraction of regularities and patterns that were implicit in the original learning experience, and selective strengthening of memories that are tagged as important, while allowing less relevant memories to decay. The optimal timing of sleep for memory consolidation appears to be within the first 24 hours after learning, with sleep occurring soon after learning providing greater benefit than sleep delayed by days. Though sleep continues to benefit memory even when delayed, and the effects of sleep on memory are most evident for complex material that benefits from integration and reorganization, rather than for simple rote associations that are already fully encoded during initial learning. Mental quietness is not the same as mental emptiness, but rather a state where cognition continues, but without the effortful, goal-directed quality that characterizes active problem-solving or task performance. And neuroimaging during meditation practices designed to cultivate mental quietness shows not absence of brain activity, but rather a shift in the pattern of activity toward default mode network dominance, decreased activation of executive control networks, and sometimes increased coherence or synchronization of activity across brain regions, indicating a more integrated, less fragmented state of processing. The subjective experience of mental quietness, when it occurs spontaneously or is cultivated through practices such as meditation or progressive relaxation, is characterized by decreased mental chatter or internal dialogue, reduced tendency to evaluate or judge experiences, decreased activation of future-oriented planning or past-oriented rumination, and a quality of present-centered awareness that may feel spacious or expansive, compared to the more constrained, focused quality of goal-directed cognition. The parasympathetic nervous system effects that accompany mental quietness include not only the previously mentioned cardiovascular changes, but also increased digestive activity and secretion, since parasympathetic activation promotes rest and digest functions, relaxation of bronchial smooth muscle, though this is typically not noticeable during quiet breathing, pupillary constriction, which optimizes near vision and reduces sensitivity to bright light, and increased lacrimation and salivation, which support tissue maintenance and repair. These various effects of parasympathetic activation collectively shift the body towards states that support recovery and conservation of resources, rather than immediate action, and the coordination of these effects through the vagus nerve and other parasympathetic pathways ensures that multiple organ systems adjust in concert to create a coherent physiological state appropriate for rest and recovery. The neuroscience of routines and habits has revealed that striatal circuits involved in habit formation show characteristic changes in activity patterns as behaviors become habitual, with initial broad activation across the entire behavioral sequence, gradually becoming concentrated at the beginning and end of the sequence, suggesting that the sequence becomes represented as a unified chunk or program that can be initiated by the starting context and then executes automatically until completion. The dorsolateral striatum, which is crucial for habitual behavior, receives dopaminergic input from the substantia nigra, and this dopamine signaling appears to provide a learning signal that strengthens the associations between contexts and behavioral responses. When behaviors lead to positive outcomes, and disruption of dopamine signaling in this circuit impairs habit formation, while leaving goal-directed action selection relatively intact. Dissociating these two forms of behavioral control at the neural level, habits can be distinguished from goal-directed actions by their insensitivity to changes in outcome value, meaning that once a behavior has become habitual, it will continue to be performed in response to its triggering context, even if the outcome that originally motivated the behavior is no longer desirable or valuable. And this insensitivity is a defining feature of habit that reflects the stimulus-response associations in striatal circuits, operating independently of prefrontal circuits that represent goals and outcomes and modulate behavior based on current values and priorities. The automaticity of habits means they can feel effortless and even compulsory, occurring without conscious decision, or sometimes despite conscious intention to do otherwise. And breaking unwanted habits typically requires either avoiding the contexts that trigger them, or effortfully overriding the automatic behavioral tendency each time the triggering context is encountered, both of which require prefrontal cognitive control and are metabolically costly and subjectively effortful, compared to simply allowing the habit to execute. Environmental design can support or hinder desired behaviors by manipulating the contextual cues that trigger habits and the ease of performing different actions. And this principle underlies approaches to behavior change that focus on modifying environments rather than relying primarily on willpower or motivation, recognizing that behavioral control is distributed between automatic, context-triggered processes and deliberate, goal-directed processes, and that the relative influence of these different control systems can be shifted by environmental manipulations, reducing cognitive load through environmental simplification, clear cues for desired behaviors, and removal of obstacles to those behaviors can make it easier to establish new habits and maintain them with less ongoing effort, compared to situations where the environment is complex, ambiguous, or contains many competing cues for undesired behaviors. The temporal dynamics of neural processing operate across multiple timescales simultaneously, with very rapid processes, such as individual action potentials, occurring on millisecond timescales, perceptual processing and attention operating on tens to hundreds of milliseconds, working memory maintenance requiring sustained activity over seconds, and long-term plasticity and memory consolidation unfolding over hours to days to weeks. And these different timescales are implemented by different neural mechanisms, ranging from voltage-gated ion channels that open and close in microseconds, to gene expression changes that may take hours to produce functional proteins, and structural synaptic modifications that may take days to stabilize. The coordination across these different timescales allows the nervous system to respond rapidly to immediate demands, while also learning from experience and adapting to longer-term patterns in the environment. Oscillatory activity in the brain, where populations of neurons show rhythmic fluctuations in their firing rates at frequencies ranging from less than 1 cycle per second to over 100 cycles per second, plays important roles in coordinating neural processing across different regions and different timescales, with slower oscillations such as delta and theta rhythms associated with sleep and memory consolidation, intermediate alpha and beta rhythms associated with attentional states and sensory processing, and faster gamma rhythms associated with local circuit processing and feature binding. The phase relationships between oscillations at different frequencies provide a mechanism for temporal organization of neural activity, with faster oscillations modulated by the phase of slower oscillations, creating hierarchical temporal structures that may support integration of information across processing stages and encoding of temporal sequences in memory. Prediction error signals, which arise when sensory input differs from expectations generated by internal models, are thought to be carried by specific populations of neurons in cortical circuits that compare top-down predictions from higher cortical areas with bottom-up sensory signals from lower areas. And these prediction error signals serve as learning signals that update internal models to better match the statistical structure of the environment, gradually reducing prediction errors over time. As models become more accurate through experience, the magnitude of prediction error signals is proportional to the unexpectedness of the input, with highly unexpected events generating large prediction errors that drive rapid learning and allocation of attention, while predictable, expected events generate minimal prediction errors and require little processing or learning. And this differential processing is one way the brain achieves efficiency, by allocating resources in proportion to the information content or surprise value of inputs. The concept of free energy in theoretical neuroscience provides a unified framework for understanding perception, learning, and decision-making as processes that minimize a quantity called variational free energy, which bounds the surprise or improbability of sensory observations given internal models, and minimizing free energy is equivalent to maximizing the accuracy of internal models while minimizing their complexity, creating a trade-off similar to Occam's razor, where the brain seeks the simplest model that adequately explains observations. This framework predicts that organisms will tend to seek out environments and engage in behaviors that match their internal models, because such environments generate low prediction errors and low free energy, while avoiding or escaping from environments that generate persistent prediction errors and high free energy. And this theoretical prediction aligns with the empirical observation that people and animals prefer familiar environments and predictable situations compared to novel, uncertain situations that require extensive model updating. Interoception, which is the perception of internal bodily states, including heart rate, breathing, digestive activity, and other visceral signals, is processed through pathways that carry information from internal organs through the spinal cord and vagus nerve to brainstem nuclei, and then to the insula and other cortical areas. And this interoceptive information contributes to emotional feelings, motivational states, and sense of self, with individual differences in interoceptive sensitivity potentially contributing to differences in emotional reactivity and susceptibility to anxiety and related disorders. The integration of interoceptive signals with exteroceptive sensory information from the external environment, and proprioceptive information about body position and movement, allows the brain to maintain a coherent representation of bodily and environmental state that supports adaptive behavior, and disruption of interoceptive processing may contribute to various clinical conditions, including anxiety disorders, where benign bodily sensations are misinterpreted as threatening. Eating disorders, where hunger and satiety signals are not accurately perceived, and dissociative conditions, where the sense of connection to bodily experience is diminished. Neural noise, which refers to random variability in neural activity that is not driven by specific signals or stimuli, is present at all levels of the nervous system, from the stochastic opening and closing of ion channels in individual neurons to variability in the precise timing and number of spikes in neural responses to identical stimuli, and this noise has traditionally been viewed as a limitation that the brain must overcome to extract signals reliably, but more. Recent perspectives suggest that neural noise may serve functional roles, including facilitating exploration of different behavioral options, preventing the system from getting stuck in suboptimal states, and allowing flexible switching between different activity patterns. The level of neural noise is not fixed, but can be modulated by neuromodulatory systems, including norepinephrine and acetylcholine, that adjust the signal-to-noise ratio in neural processing, with higher neuromodulator levels generally increasing the relative strength of signals over noise and supporting focused processing, while lower levels allow more noise and support more diffuse, exploratory processing. Metabolic constraints fundamentally shape neural architecture and function because neurons are among the most metabolically demanding cells in the body, with the high energy costs coming primarily from the need to maintain ionic gradients across cell membranes against concentration gradients, and these gradients are necessary for generating action potentials and releasing neurotransmitters. And every spike that a neuron fires requires ATP to pump ions back to their resting distributions, and this energy cost means that neural coding strategies that use fewer spikes to represent information are metabolically advantageous compared to strategies that require high firing rates, and this cost constraint has likely influenced the evolution of neural codes towards sparse representations where only a small fraction of neurons are active at any time, rather than dense representations where most neurons participate in every computation. The glial cells that make up approximately half the cells in the brain, including astrocytes, that support metabolic functions and maintain the chemical environment around neurons, oligodendrocytes, that form myelin sheaths around axons to speed signal transmission, and microglia that serve immune functions, are essential for normal neural function, though they do not themselves generate action potentials or directly participate in information processing, and these non-neuronal cells outnumber neurons in some brain regions and play crucial roles in neurovascular coupling that adjusts blood flow to match local metabolic demands, in buffering neurotransmitters and ions in extracellular space, in providing metabolic substrates to neurons, and in synaptic plasticity through release of gliotransmitters and modulation of synaptic structure. The blood-brain barrier, which is formed by tight junctions between endothelial cells lining brain capillaries and is reinforced by astrocytokine feet surrounding the blood vessels, restricts movement of molecules between blood and brain tissue, protecting the brain from toxins and pathogens in the blood, while also creating challenges for drug delivery, since many molecules that could have therapeutic effects on brain function cannot cross this barrier, and the selectivity of the barrier means that the brain has specialized transport systems for glucose, amino acids, and other essential molecules, and the barrier can become compromised in various pathological conditions, including inflammation, trauma, and some neurodegenerative diseases, potentially contributing to disease progression through allowing entry of blood-borne molecules that are normally excluded from brain tissue. Sensory adaptation, where neural responses to constant stimuli decrease over time, is a ubiquitous feature of sensory systems that allows them to remain sensitive to changes in stimulation rather than being dominated by constant background levels, and this adaptation occurs at multiple levels, from receptor cells that show decreased responsiveness to sustained stimulation, to cortical neurons that adjust their response properties based on the statistics of recent input, and the time course of adaptation ranges from very rapid adaptation occurring within milliseconds to slow adaptation that develops over minutes or hours of exposure, and these different timescales of adaptation allow the system to adjust to both transient and sustained changes in the sensory environment. The perceptual consequences of adaptation include phenomena such as the decrease in perceived brightness of a room after entering from outdoors on a sunny day, the fading of awareness of clothing touching skin after wearing clothes for a few minutes, and the reduction in odor perception after prolonged exposure to a smell. And all these examples illustrate how sensory systems prioritize detection of change over absolute levels. Motor learning proceeds through stages, beginning with a cognitive stage where performance is slow and effortful and requires conscious attention to each movement component, progressing through an associative stage where movements become smoother and more consistent, though still requiring some attention, and reaching an autonomous stage where movements are rapid, efficient, and require minimal conscious monitoring. And these stages correspond to shifts in neural representation. From widespread cortical activation during early learning to more focused activation in motor areas, and eventually to involvement of subcortical structures, including cerebellum and basal ganglia, that support automatic skilled performance. The cerebellum, which contains more neurons than the rest of the brain combined, though they are smaller neurons, densely packed in the cerebellar cortex, is critical for motor timing and coordination, and for learning to adjust movements based on sensory feedback about errors and damage to the cerebellum, produces characteristic deficits in movement coordination, balance, and motor learning, while leaving other cognitive functions relatively intact. Error-based learning, where performance is adjusted to reduce differences between intended and actual outcomes, is implemented in the cerebellum through climbing fiber signals that convey error information to Purkinje cells and modify the strength of synapses between parallel fibers and Purkinje cells, and this modification allows motor commands to be updated to compensate for errors, and eventually to anticipate and prevent errors through feed-forward control and similar. Error-based learning mechanisms operate in other brain circuits, supporting various forms of procedural and skill learning. The consolidation of motor skills shows a time course similar to declarative memory consolidation, with initial learning followed by a period of offline gains where performance improves during rest or sleep without additional practice, and these offline improvements are associated with neural replay of activity patterns that occur during practice, suggesting that consolidation involves reactivation and strengthening of task-relevant neural circuits during periods of rest. The ventral and dorsal visual streams, which diverge from primary visual cortex and project to different parts of the brain, are often characterized as serving what and where, or what and how functions, with the ventral stream projecting to inferior temporal cortex, supporting object recognition, and the dorsal stream projecting to posterior parietal cortex, supporting spatial processing and visually guided action. And these streams process different types of information from the visual input, with the ventral stream sensitive to object features such as shape, color, and texture, while the dorsal stream is sensitive to spatial relationships, motion, and the relationship between objects and the observer's body. Damage to ventral stream areas can produce agnosia, where patients cannot recognize objects despite intact basic vision, while damage to dorsal stream areas can produce spatial neglect, where patients fail to attend to one side of space, or optic ataxia, where visually guided reaching is impaired despite preserved object recognition. And these dissociations demonstrate that what we experience as unified visual perception depends on multiple parallel processing streams that can be selectively disrupted. Statistical learning, where the brain automatically extracts regularities and patterns from sequences of events, even without explicit instruction or awareness, occurs across multiple domains, including language acquisition, where infants track statistical patterns in speech sounds to segment words from continuous speech, visual processing, where repeated co-occurrences of features are detected and used to facilitate object recognition, and motor sequencing, where temporal regularities in action sequences are learned implicitly, and this learning appears to engage corticostriatal circuits similar to those involved in habit formation and procedural learning, and the learning proceeds gradually through exposure to many examples rather than through sudden insight or hypothesis testing, and the learned patterns can influence behavior even when people cannot explicitly state what they have learned, indicating that statistical learning operates largely outside conscious awareness. Attentional blink refers to a temporary deficit in detecting or identifying a second target stimulus when it appears shortly after a first target in a rapid sequence of stimuli, with the blink typically lasting 200 to 500 milliseconds after the first target, and this phenomenon demonstrates the temporal limitations of conscious perception and indicates that consolidating a stimulus into working memory requires sustained processing over several hundred milliseconds, during which the attentional system cannot be fully deployed to other stimuli, and individual differences in the magnitude of the attentional blink correlate with differences in working memory capacity and attentional control, suggesting that the blink reflects fundamental capacity limits in the systems that support conscious perception and short-term memory. change blindness, where people fail to notice substantial changes in visual scenes when the change occurs during a brief interruption, such as a saccadic eye movement, or a blink, or a cut between images, demonstrates that visual perception is not a complete, detailed representation of the visual field, but rather a sparse representation of attended objects and relations, with much of the detail filled in based on expectations and general knowledge, and this finding has implications for understanding eyewitness testimony, user interface design, and the relationship between perception and reality, showing that we are much less aware of visual details than subjective experience suggests, and that attention is necessary for encoding changes into memory, even when changes are clearly visible. The binding problem refers to the question of how the brain combines information from different processing streams and different neural populations to create unified perceptual experiences of objects with multiple features such as color, shape, location, and motion, all processed in different brain areas, and proposed solutions to this binding problem include temporal synchronization, where neurons representing different features of the same object fire in synchrony, thereby tagging those features as belonging together, attentional mechanisms that selectively enhance processing of all features at a particular location, and convergence zones, where information from different processing streams comes together in higher-level areas that represent conjunctions of features, and likely multiple mechanisms contribute to binding, with different mechanisms operating for different types of features and different task demands. The nervous system's representations of sensory stimuli, motor actions, and abstract concepts are distributed across populations of neurons rather than being localized in individual neurons or small groups of neurons, and this distributed coding provides robustness to damage or noise, since the loss of individual neurons does not completely eliminate information, and distributed codes can represent more distinct items than local codes, where each concept is represented by activity of a single neuron, and population coding schemes can represent not only identity of stimuli, but also uncertainty or confidence through the pattern of activity across the population, with sharp, concentrated activity patterns representing high confidence, and broad, distributed patterns representing uncertainty. Neural representations are not static, but change over time through learning and experience, with repeated exposure to particular stimuli or tasks, leading to changes in which neurons respond to those stimuli and how strongly they respond. And these changes reflect plasticity mechanisms that optimize neural codes for behaviorally relevant information and for efficient use of neural resources. And plasticity is not limited to early development, but continues throughout life, though the rate and magnitude of plastic changes generally decrease with age, and maintaining some plasticity into adulthood allows ongoing learning and adaptation to changing environments, though excessive plasticity in mature circuits could destabilize established functions and memories. The overall pattern of neural organization reflects a hierarchy from primary sensory and motor areas through secondary and association areas to multimodal and executive control areas, with representations becoming progressively more abstract and integrated across modalities at higher levels of the hierarchy, and this hierarchical organization allows complex processing to be decomposed into simpler operations distributed across multiple processing stages and feedback. Connections from higher to lower areas allow top-down influences, including attention and expectation, to modulate processing at early stages, creating an interactive system where both bottom-up sensory information and top-down context influence representations at every level. And now, as everything gently fades into stillness, as the universe breathes in quiet harmony, there is nowhere you need to go, nothing more to seek, nothing to hold. This is enough. You are enough. Let the silence cradle you. Let the darkness feel safe and soft. Let your thoughts dissolve like stardust. May your body sink into rest. May your breath become slow and easy. May your dreams open gentle doors to peace. As the cosmos continues its endless motion, you may drift effortlessly into sleep. And when morning comes, may you rise, calm, renewed, and light, carrying a quiet echo of the infinite. Good night, and rest among the stars.